Good morning, and thank you everybody for being here. I would like, how do I change the... Okay, yeah, it's difficult. Okay, thank you. All right, now we sorted this out. So good. So good morning again. Good morning. And thanks for being here and for giving me the opportunity to present you Immunicum and give you an update on the exciting times that we have for the company, as you saw in the last days in terms of announcements. But let's first talk about what is Immunicum. We are a clinical stage, a biopharmaceutical company, and we really thrive to develop and establish a unique immuno-oncology approach. And this is done through allogeneic and off-the-shelf product. And I will explain what does this mean, because it's very important in the very crowded field of immuno-oncology that we as a product are allogeneic, means that it's derived from healthy donors, and off the shelf the product is ready to be administered sitting in the hospital. So we basically, our goal as a company, and myself also as an individual, as a physician, has always been to try to make a difference in patients' life, and in this case to increase survival and improve quality of life. Our lead product, ilixadencel, as you will see, are pro-inflammatory allogeneic dendritic cells, and basically our objective as a company is to make ilixadencel a core part of the combination treatments for cancer. That is today really the goal. Everybody accepts that if you want to treat cancer, it's not one single product that will do it. You need at least a combination of two, sometimes even more. We are listed in the Nasdaq First North, and now really in the last stages of moving to the main market. As you probably saw in our announcement on Friday, the Nasdaq committee has given a conditional approval. This is the first time that to do that. Basically, we fulfill all the requirements as a company to move to the main market, and the only thing that we need is now next Monday, when we have a EGM, to approve the financing, and that is the only condition that is left. And with that, we expect to be uplisted and move to the main market small cap in January. This is our pipeline. We have 3 platforms, 2 in preclinical and 1 in clinical. That's where I will spend most of the time. And we are currently in phase 2 in kidney cancer, and we just finished in liver cancer. And we also have a collaboration with the Karolinska Hospital for a rare disease, gastrointestinal stromal tumors. Only 200 cases a year in Sweden, but patients with very big needs and not a lot of alternatives for them. But let me tell a little bit about where are we in terms of the immuno-oncology is now probably the fastest growing segment in all of pharmaceuticals, not just cancer. And what is immuno-oncology? Basically, immuno-oncology, we are trying to use the patient's own immune system to attack the cancer cells. You can do this from 2 different approaches. One is to block the defense mechanisms that the tumor cells create against the patient immune system. That's one approach. The other one is to activate the patient's own immune system so that they can be stronger and in bigger number and attack and kill the cells. Most of the growth in the last couple of years have been on the side of blocking the defenses that the tumor cells create with the checkpoint inhibitors. As you can see, huge growth, and all in the last couple of years. 17 billion just in this sector in 2015, and expected to grow in 2022 to 76 billion. So huge, expectations in terms of the innovation in all of this field. So when you look at the survival, you see here the green line is the typical with chemotherapy. Then with these new therapies, the checkpoint inhibitors, you increase the survival of these patients in monotherapy. But now everybody accepts that if you want to further increase the survival, you need to do combinations. And the reason is these checkpoint inhibitors have limitations. So the tumor cells don't all the time are blocked from blocking the immune system. So you need to complement that efficacy. So this is what I'm showing here. So this is the cancer immunity cycle. Here you see the tumor cells. And basically, as I mentioned, they create these defenses. They block the immune system to attack the tumor cells. And this is where these classes, the checkpoint inhibitors and the TKIs like sunitinib, attack. But you can block the defenses. But if the patient immune system is not strong enough, is not active enough, it really doesn't matter. I normally compare it with a castle. The castle is a tumor. So, you know, the checkpoint inhibitors and the TKIs destroy the gate, the entrance to the castle. But if your troops are not strong enough and in big number, it doesn't matter because you cannot go and destroy the castle. So you need troops that are really strong and activated. And that's what we do on the side. We prime, we activate the patient immune system to attack the cancer cells at the site of the tumor, but all over the body. And I will tell you how that happens. So one of the key differences of our product versus other primers is that, as I mentioned to you, we are originated from healthy donors. So one healthy donor gives about 100 doses. In kidney cancer, we treat with 2 doses, so one donor treats 50 patients. And this is a big difference to other cell-based therapies, that the patient is the source of the vaccine, because you need to do biopsy, select it outside of the body, and then re-inject in the patient. Very time-consuming, very expensive. So this is very practical. We have a batch of 100 vials that we ship around. The product sits in the hospital, just like this, a little vial. And then when you need it, you just put it at body temperature and inject into the tumor. So in terms of the cost of manufacturing, has no impact in the margins. So very importantly, when you want to activate the patient system, is that now is accepted that you should use the tumor as the source of the neoantigens. The neoantigens are, think about it as barcodes, very specific to that tumor in that patient. So you want to be able that the patient's immune system recognizes that barcode and goes around the body, and when it sees the barcode, destroys the cells. So when you inject into the tumor, we are going, by the nature of our product, these cells that are made very active with a cocktail are going to recruit the patient immune cells, called dendritic cells and natural kill cells, to the site of the tumor. So you bring more cells than what you normally will see there. These cells are going to kill the tumor cells and release these codes. So you have then the dendritic cells that are from the patient ones that with our cocktail become very activated. They take on these codes, they go to the patient lymph nodes and train then the immune cells called natural killer T cells that then go spread through the body and attack the tumor cells. So it's very important that you bring into the site of the tumor the patient's immune cells in big quantity, that you train them to recognize that specific barcode, and that you activate them so that they train the killer T cells. And those are the most important cells that to really kill the tumor cells. So this is everything we do with our product, and the bigger differentiation with other ones. Very exciting. We have had recently, published preclinical data where we showed this mechanism of action. How do we activate the patient's immune system? But even more exciting for us is that today, this morning, so probably some of you haven't seen, we have preclinical data where we show that when we combine our product, ilixadencel, with the checkpoint inhibitors, you get a much stronger effect on the survival. This is, of course, done in animals, in mice. And as an example, at day 24 after treatment, the mice that were treated they had the tumor and were treated with the checkpoint inhibitors alone, at day 24, none of them was alive. So when they were treated only with our product, 30% of them were alive. When you treat them in combination of the checkpoint inhibitor plus ilixadencel, 50% of them were alive. So you see here that the checkpoint inhibitor practically no impact. Ilixadencel by itself had already a good impact, but the combination then really made an impact on the survival of these mice. And of course, we cannot extrapolate from mice to humans, but this is just to explain the rationale of combining these two products and the first preclinical data that we have in combination of ilixadencel with checkpoint inhibitors. And as I will explain to you, this is the next study we want to do, because that's a lot of the future. And now, you can see the details in the press release, and then we will be analyzing the data. Some mice are still alive, so we need to continue following them, and then we'll make a publication. But I think more exciting, of course, this is exciting for us, this data, but the most important for us is the data when we use the ilixadencel in patients. This is our first clinical trial. We published the results in June in a very prestigious journal, the first publication that we had, clinical publication. And here, these patients had kidney cancer with metastasis, and they were newly diagnosed. So normally what you did, you injected into the tumor one day, 2 weeks later you injected a second dose, and then normally 2 weeks later you remove the kidney that has the tumor. And if the patient progresses, then you treat them with the standard of care. And what you see here is the last time we look at the survival, is 5 out of 11 patients were still alive. Passing already the 4-year mark, one of them the 5-year mark. So 48 months was the median overall survival in the study. With the standard of care alone and historical data, the median overall survival is 15 to 16 months. So it's a small number of patients, but in terms of the impact on this patient's life, again, 5 out of 11, and extending from 15 to 16 to 48 months is impactful in this patient's life. So very good data, of course, supporting the other studies that we are doing. And this publication has been very useful when we talk with the investors and potential partners. This is just to show you, in terms of one of the patients in that study and why we say that the combination is the preferred way of moving. This patient had the kidney cancer, then had the liver metastasis, lung metastasis, and then he developed 4 brain metastases. And normally, you don't expect much, there's only one outcome here. So anyway, he was treated with sunitinib, Sutent, that is the standard of care, because that was the only alternative, although it's known that Sutent doesn't do anything to the brain metastasis because it doesn't pass the blood-brain barrier. So just to tell that this patient then started improving, the brain metastasis went away, the liver metastasis went away. Last December, he had 1 small lung metastasis that was removed surgically. So he was still alive 48 months after being treated, and he had no metastasis. He lost his driver's license here, he got it back here. And here's just to say that although Sutent doesn't get to the brain, reduces the number of tumor cells in the whole body and gives opportunity for the patient immune system to then go and attack these tumor cells. So the benefit of the combination. And this is just to show you what you can see of this concentration. The dark is these cells that I mentioned to you, the killer T cells, very specific to the tumors. This is a tumor that was not treated. You see some little dots. This is a normal reaction. And all these images are from one of those patients. You see the massive infiltration in the tumor of these T cells, killer T cells. Nothing on the kidney tissue around, very specific to the tumor, but also on the metastasis. These cells are not just in the tumor, but they travel through all the body and also attack the metastasis. So where are we now? After this study, we initiated a phase 2 study That means it is a controlled study, 90 patients comparing ilixadencel plus the standard of care, sunitinib, versus the sunitinib alone. This is the most important type of data that you can produce to authorities and potential partners, comparative data, where you can really see the benefit between the groups. So out of a total of 90 patients, we have now 80, and we expect to close enrollment by the end of the year. And this study is running in 8 European countries and in the US. And was also very important to get approval by the FDA to run studies in the US, because it's also not that easy. And so we expect, as I mentioned, to close enrollment at the end of the year, and then follow these patients with the top-line results in 2019. The GIST study, again, very slow enrollment because it's a rare disease, but we continue moving. It's very important for these patients. And we also just recently also announced the top-line results in a liver cancer study, phase 1/2, very terrible disease, very fast progressing. But basically here we confirmed also the safety of our product. So one of the problems with a lot of these immuno-oncology approaches is that the toxicity, the safety, these treatments are impact on patients' lives. With ilixadencel, we have now more than 85 patients treated, and the big side effect that we got was fever, what you would expect because you induce a reaction, and was treated with paracetamol. So very, very safe. What is ideal to combine with classes of drugs that are not the most well tolerated. So I showed you all this, and you will have access to the presentation, because I want to move now to the next phase of our development. So we have now going to have data in kidney cancer, in liver cancer, with one class of drugs, the TKI, sunitinib, but now we want to move into the big development and a lot of the innovation combination with checkpoint inhibitors. And this is the study we are already planning to initiate next year, and looking at these indications that I will tell you why. We want to finish the current studies and also prepare the product when the phase 2 study to initiate pivotal studies. For that, you need to develop what we call a commercial process in terms of manufacturing. And this is everything we want to do with the money that we are raising currently. Why did we pick up these indications? These indications are very big. Head and neck cancer, gastric adenocarcinoma, non-small cell lung cancer. Very big indications where the checkpoint inhibitors alone have relatively poor efficacy. So you don't need a lot to show a difference. And also, where you can recruit patients was very important in this field, is that you can recruit patients. So this plan is also going to reduce the development risk for the company, because now you have kidney cancer, liver cancer, and another 3 cancers and 2 classes. So, as a company, we are diversifying the risk and reducing this risk in terms of development. So very, very important. All these indications are big in terms of the potential. And here is what we are raising. So we already have guaranteed 200 million SEK out of a total of 223 we want to raise. As I mentioned to you, we are going to have an EGM next Monday for approval of this financing. And the only conditions that we are required to perform is that the EGM approves, of course, the raising the money, and that the authorities approve the prospectus. Some people sometimes confuse that if the subscription price, that is 8 krona, comes down, the guarantees can exit. It's not true. So the guarantors are committed to financing regardless of the share price, but we are above that. So the only 2 things we need is EGM approval, and we can execute this, and the prospectus being approved. And when we get this approved, then we will get the Nasdaq committee approval to move into the main market, what is very important for the company. So this is the details about the Nasdaq feedback, committee feedback. Basically, we just need to do this. And a series of news that are going to come during next years. With the new study, of course, they are listing to the Nasdaq, and we expect to get the initial results in terms of safety and for the new study. And then 19 is a very strong year in terms of data, because we are going to get the data from the Mareca, from the GIST, and some initial indication of efficacy in combination with the checkpoint inhibitors. So I hope that this gave you, you know, a little bit of a summary of the company, that we have quite a unique approach to activate the patient's immune system, that we have really, because of this profile and the safety, very nicely positioned as a backbone in the treatment, future treatment of cancer in combinations. Our advanced clinical data supports moving forward. Also, the safety profile that is very good, supports again the combinations, and also we know this is the fastest growing. This is the field with a lot of innovation, and we are really at the very front in the group of the primers. So thank you so much.
Thank you, Carlos. Thank you. I was thinking there has been a few disappointments when trying to develop cancer vaccines. Bavarian Nordic failed its phase 3 study, is one example. But why do you think you stand a better chance of going the whole way?
I think you know, one of the key differentiation we have with some other vaccine approaches is that we use the allogeneic approach. You know, where a lot of the other companies use the patient as the source of these neoantigens, so that you need to select them out externally, ex vivo, and then you need to inject back, and then did you select the right ones or not? We don't need to do any selection, because the tumor is the source of these neoantigens. So that already makes sure that when you inject it, it's going to be personalized for that patient and for that tumor. I think in addition, you know, we have very good data showing the mechanism of action. How do we interact? How do we activate the patient immune system? And there's been, you know, every month we come with new data strengthening that aspect. The mode of action, now we have this preclinical also showing how do we interact with the checkpoint inhibitors, very important, supporting the new clinical trial. But I think, you know, overall, considering that we already have data in 2 types of cancer, and you know, an extended survival in the kidney cancer, I think there is already some data that at least points that we are following the right approach. Of course, this is development, this is biotech. There is always a risk, but I think that when you look at the number of companies operating in this area, we are quite ahead, and you know, and the more you move, the less is the risk. So yeah, within the risky part.
Yeah, like you mentioned, when looking at the clinical data, there's the survival rate is of course very encouraging, but the effect on the cancer tumor itself has not been as apparent. Why do you think we see this paradox, so to speak?
You're talking in general, or
yeah, in general?
No, I think you start seeing definitely more proof that the immuno-oncology approach, it works. You know, I'm not talking now about the non-solid tumors like you know, the blood cancers now with the CAR-Ts that have been making a big difference. But if you look at the checkpoint inhibitors in some cancers like melanoma, skin cancer, has been a big impact. You know, it has an efficacy of 70%. I think in other cancers they're becoming standard of care. You start getting patients that respond. I think the challenge that they are facing, and the reason why we move into combinations, is that cancer cells are very smart cells. So you cannot just have one single approach to attack the type of tumors. So that's why different combinations are important. And I think that is now going to be the future. Most of the data that you have so far has been with monos, just the single product, the checkpoint inhibitors. You are going to start more and more to get data on combination. You start seeing this difference. So you start getting more and more data showing really the impact of priming the immune system to attack the cancer cells.
Okay. And you announced quite a large share issue. Will the management subscribe to the issue?
Yeah, absolutely. All management will subscribe. The new members in the management that are not yet shareholders, because for many months, until quite honestly until today, we were in a black period. No one could buy or sell shares since February. So the new management that joined the company could not buy shares, but they also want to participate. So myself, I will participate pro rata. So everybody is behind the product and the company.
That's good. I mean, your last share issue, those who invested in that and held on to the share has not seen that great of a development on their portfolio. But why do you think it's a better opportunity now to do it than?
I think, look, some people ask me, what are you doing about the share price? I really cannot do anything about the share price. I don't control I don't control Nasdaq, and I don't control the share price. What I think that as a management, what we have been delivering, if you look at how the company was at the last time of the last share issue, so what have we been delivering from that time? We got the FDA to let us run studies in the US. We got the first clinical trial publication. We got now, pretty much concluded the move into the main market. We produce additional preclinical data showing the mechanism of action and the benefit of combining with checkpoint inhibitors. We got the first data on the liver cancer. So, I can understand the disappointment about the share price, but I can say that as a company, we are in a much stronger place today than what we were 1.5 years ago. And I was talking with some specialist investors, and they say, I don't care about the share price today. What I care is what this money that you are going to use is going to deliver in terms of the program, and what is going to be the share price at that time. So I think that the only thing I can say is that we continue to deliver on what we committed to deliver. I think the company is in a much stronger place. That is a reflection, the fact that we could secure 200 million, what is not easy in Sweden, and that the whole management and the board is behind this transaction and the plan to move forward.
Can you tell how much you paid the guarantors?
That will all be disclosed, next a week from today, in the prospectus and the conditions. Standard,
Yeah. Do we have any questions from the audience? No questions from the audience? Then I'll move forward to your new strategy. You will target more types of cancers. Can you give us a hint of which cancer form you see the most potential?
I think in the oncology space, it's important that when you start getting results in different types of cancers, this moves to other types of cancer. So what is important for us is to prove the concept that when you use ilixadencel and the checkpoint inhibitors together, you are going to get a better result. So we selected these 3 cancers because, as I mentioned, they are big. A lot of patients don't respond to them, or then they relapse. So, there is a patient need in these cancers to be treated with different approaches, in this case, the combination. Also very important in this field, because it's so busy, so innovative, that some types of tumors are very crowded in terms of selecting and recruiting patients. We selected these tumors because you still can recruit patients. And we also selected tumors where the checkpoint inhibitors are already being used to treat these patients, so that it's easy for us to combine with them. So when we go into 3 types of tumors, and in this study we'll have 3 arms, one of each in each tumor. So we are multiplying, what I call the shots, because also very, very easy, as I said, towards the end of 19, we are going to get this first checkbox, that is only going to be done with 7, 9 patients. And you say, if 1 out of, as an example, if 1 out of 9 gets this response, you can continue. If not, then you stop. So, so there is you increase the chances of moving, versus doing first one, and then that is not a good one. Then you go and you lose valuable patent protection time. That is very important in this field. So we spent some months, but we spoke to, internally, but with a lot of experts, both in the US and in Europe, key opinion leaders, practitioners, the organizations that run the studies. Inclusive, we spoke to one of these organizations in Australia, because people said, it's easy to go into melanoma. We talked with everybody, and they all told us, including in Australia, you will not be able to recruit patients in melanoma. So we just confirmed that. So this is not just an internal discussion. We spend months talking with the experts in the different areas and say which ones makes sense for us to go.
Sounds interesting, Carlos.
Thank you.
But our time is out. So thank you very much.
Thank you.